Welcome to the audiobook Biology 11th class chapter 2 Biological Classification 2.1 Kingdom Monera 2.2 Kingdom Protista Kingdom Fungi Kingdom Plantae Kingdom Animalia Viruses Viroids and Lichens. Let's start. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification system for the living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of the other kingdoms have also been understood differently by different scientists over the time. Table 2.1 Characteristics of five kingdoms. Five kingdoms. Characters cell type, cell wall, nuclear membrane, body organization, mode of nutrition. Cell type in Monera is prokaryotic, protista eukaryotic, fungi eukaryotic, plantae eukaryotic, animalia eukaryotic. Cell wall in Monera is non-cellulosic polysaccharide plus amino acids. In protista, present in some fungi present with chitin, plants present cellulose, and in animalia cell wall is absent. Nuclear membrane in Monera is absent. In protista present, fungi present, plantae and animalia present. Body organization in Monera is cellular. In protista it is also cellular, but from fungi it is multicellular but loose tissue. In plantae tissue organ, and in animalia tissue organ system. Mode of nutrition in Monera is autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic, parasitic, all kind of. In protista, autotrophic, photosynthetic, and heterotrophic both. In fungi, heterotrophic, saprophytic, and parasitic in nature. And in plantae, autotrophic, photosynthetic. And in animalia, it is completely heterotrophic, holozoic, saprophytic, etc. R.H. Whittaker in 1969 proposed a five kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain, and thereby a six kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five kingdom classification to understand the issues and considerations that influence the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organisms and multicellular ones. Say, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group, fungi, and the autotrophic green plant, though they also showed a characteristic difference in their walls composition. The fungi had chitin and in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae within plants and both having cell walls, with Paramecium and amoeba, which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over the time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic. This is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt separately in chapter 3 and chapter 4. 2.1 Kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans, where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus, plural is cocci, the rod-shaped bacillus, plural is bacilli, and the coma-shaped vibrium, plural is vibrio, and the spiral spiralum, plural is spirilla. Figure 2.1, bacteria of different shapes, cocci, bacilli, spirilla, and vibrio. Though the bacterial structure is very simple, they are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic. It implies they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs. It implies they depend on the other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals.